Oh, thank you very much for inviting me again. First, I would like to say how grateful I am to San Francisco State for really getting me moving in my career. Without them, I'm not sure where I would be now. So today I'm going to talk about targeting prostate cancer, the moving target. I've been thinking about precision medicine for many years now, in fact, since I was on Dean Kessler's committee on precision medicine at UCSF back in the early 2000s. So this is the promise of precision oncology, that we'll do all these genomics, all these omics, and then target the tumor and kill it and cure the patient. The problem of precision oncology, though, is that it's really predict and pray, right? We're actually not doing very well in this realm right now. And so why is this so hard? So we can look at prostate cancer for lessons, because this is a fairly well understood disease now with great biomarkers and very good drugs. So globally, there's 900,000 diagnoses per year, and many men can benefit from no treatment, but we don't know how to stratify those guys, right? And in terms of risk of metastasis, and that is precision medicine in the sense that you can tell guys, you don't need therapy. Now there's nearly 250,000 deaths despite having one of the best biomarkers and second and third generation targeted and even combinatorial therapies that hit the drivers dihydrotestosterone and the androgen receptor. So we just also hit the major buzzwords of precision oncology, right? And yet 250,000 guys are dying every year. And this is a disease with marked discrepancy in both prevalence and mortality amongst different ethnicities. So Asians have very low rates, whites have intermediate rates, and African Americans have very high rates of both prevalence and mortality. So this is the sort of natural history of the disease and where the problems lie. Here, early detection, where you might get PSA or positive digital rectal exam, many of these guys can avoid any therapy at all with what's called active surveillance. But as if PSA continues to rise, then they might get a therapy. It will fall. It will then recur, invariably recur. And this is where the new therapies are quite effective in lowering disease burden again. But so these are the two problem areas, right? What you do when you have early detection with guys with low risk cancer, and then treatment of the high risk cancer, which is aggressive and generally fatal. So central to the problem one, the early detection, is this intra-prostate and intra-tumor heterogeneity. So what I'm showing you here is the grading scheme, and this just means basically the degree to which it's de-differentiated from well-differentiated to a mess. And then so these have different grading categories, so 4 plus 4 would be bad, 3 plus 3 is okay, 3 plus 4 is not so good, and the risk of post-treatment recurrence is shown down here. So for low grade, it's quite low, and for high grade, it's high, of course. And again, many men can elect to live with a cancer if it's low risk, but right now, it's hard to be certain of who can elect active surveillance and who should not. And so this is really a story of serendipity. We derived an expression signature that's predictive for metastasis, competent primary tumors, using interspecies transcriptomics. So my good friend and collaborator, Dr. Y.Z. Wang, made 5 different mouse models, right, by taking 5 different biopsies from a single prostate and putting them in mice, and because he wanted to functionalize the heterogeneity, right? And sure enough, some of these are highly metastatic, H. One is not metastatic at all, and some of them sort of intermediate. So we sequenced these genomes. These are human tumors. We sequenced these transcriptome, genome. We did everything, thinking we would find a mutation or something that would predict metastatic behavior. We found nothing. So two of our postdocs put their heads together and said, well, maybe it's something else. Now, because these are human tumors growing in mice, we could do an interesting experiment. We could analyze the stroma, the tissue that supports the tumor. When we did that, we discovered that the mouse genes, right, in the tissue supporting the tumor, perfectly segregated the metastatic and non-metastatic tumors and other mice carrying these tumors. And not only that, when we converted the mouse genes to human genes, and we went and looked in very large databases, for example, of men from the Mayo Clinic, we found that we could also predict metastasis in those guys. And even better, when we went to intermediate grade prostate cancer, where the big problem lies, do I or don't I have therapy, we found that we got even better separation. So this is crazy, but it turns out that the stromal expression signature may better stratify for risk of metastasis than tumor signatures and aid with physician-patient decision-making. And it has another benefit, because when you do biopsies, you're punching holes in the prostate, mostly blindly, looking for tumor, and you can miss the tumor, or you can get very small amounts of the tumor. But with this stroma, there's a field effect, a cancerization, as it will, around these different lesions. And so suddenly, if you're hitting near the tumor, you're doing well enough. And we think, but we don't know, that it might not matter where in the prostate you actually hit. Hopefully we'll get there soon. So when you think about all these big tumor sequencing efforts that have been going on, looking for drivers of metastasis and drivers of phenotype, maybe we've been missing half the story. So this is something we just published, and it's very interesting to us. So the other problem 2, is the problem of progression and the challenge of precision oncology. So here we have to look at a little bit of biology. So prostate cancer is driven by the androgen receptor and its ligand, dihydrotestosterone. So basically, this molecule comes into the cell, binds the androgen receptor, which dimerizes, it then goes to the nucleus and turns on a whole bunch of genes, including PSA. This is called the androgen signaling axis. Okay, now we've got great drugs now that block both production of dihydrotestosterone, abiraterone, and we've got drugs that block the binding site of the androgen receptor, such as enzalutamide. Okay, the problem is, invariably, the cell figures out how to work around this blockade. And so it can do it by amplifying the receptor. It can mutate the receptor so it becomes promiscuous and, for example, binds progesterone or estrogen. You can get mutations and things like PTEN, or it can just turn into a different kind of cell. And so this just sort of summarizes what I just told you. You treat the adenocarcinoma, the primary prostate cancer, with these strong drugs, and about 70% of the time it'll turn into what's called castrate-resistant prostate cancer, which is androgen receptor positive because it reactivates that signaling axis, or it turns in about 20 to 30% of the time into neuroendocrine cancer, which is AR and PSA negative, but it expresses neuronal markers. And this is a completely different kind of cell. So it turns out that until very recently, we didn't know anything about metastatic prostate cancer, whether it was this type or this type. And that's because biopsies were expensive, painful, and didn't really change treatment. So why do it? And a few years ago, my lab decided to see if we could develop what are called liquid biopsies. So we could just do a blood draw and analyze circulating tumor DNA to get at what's going on in patients. And this is obviously inexpensive, non-painful, relatively non-invasive. And so a team at the Prostate Center, Stas Volek, Alex Wyatt, and Kim Chi, the oncologist, we worked together to sequence the ligand binding domain of the androgen receptor and do whole genome analysis using comparative genomic hybridization, which measures copy number across the entire genome. And what we found when we looked at men pre-treatment, this is amazing. This is DNA taken from blood with guys with prostate cancer. So this is baseline here pre-treatment. And I can tell you that these copy number changes look exactly like prostate cancer, okay? And the gains and the losses are shown in blue and red, and this is like a barcode that I recognize, and this is prostate cancer. When these men went on drug, the cell-free DNA that we were measuring from the blood dropped because the drug works. And you can see that, we went from however many patients this is, we could make measurements in to much fewer because those guys were having a good response. But as they progressed, right, and their PSA started to rise again, you can see that now we have many, many more changes that we had in the primary setting, in the pre-treatment setting. So this tells us that quickly, and this is in months, okay, so this tells us quickly the tumors are evolving. And because we had sequenced the ligand binding domain and other genes, we were able to look at, what the impact of pre-existing, for example, gains of the androgen receptor, amplifications of the AR receptor, or mutations had on disease-free survival. And so what I'm showing you here is the status of the androgen receptor in red here. Red means amplification or various mutations here. And these are changes, copy number changes in other genes. And so you can see that if at treatment you have these gains or mutations, you do much worse in terms of disease-free survival, or we can think of it that way, than if you don't have many changes, in which case you do very well. And the interesting thing here is that there tends to be mutual exclusivity between gains in the androgen receptor and mutations in the androgen receptor. And in some of these men, we see up to 3 or 4 mutations, suggesting multiple metastatic tumors. So here's the scary part, right? So this is looking at men at progression and comparing their mutation status and copy number status to baseline. And this is a busy slide, and all you need to pay attention to is these arrows that are pointing up or down. These are all changes in the mutational status or copy number status since treatment began, right? And again, this is happening over the course of months, right? And I reviewed a paper recently where they were seeing these changes in only 4 weeks. So this is terrifying, okay? And what it means is that we're dealing with a dynamic ecosystem and not a tumor, right? We tend to think of it's a tumor. It's not a tumor, right? We know in some of these guys, there's multiple Mets. We know that from imaging, but here we see that they're genomically quite dissimilar in some cases. So my colleague at the Vancouver Prostate Center, Artem Cherkasov, walked into my office and said, if you give me the mutations you have, I'll functionalize them. And so we did that. We functionalized all of them. But I'm giving you an example now of a patient who came in. At baseline, he had two haplotypes, so presumably 2 detectable metastases, right? So these are sequence of the androgen receptor ligand binding domain, and the mutations are shown here. They're roughly 50-50. At progression, he presented with 1, 2, 3, 4, 5, 6 haplotypes. So presumably 6 different, genetically different metastatic tumors. So amongst other things, this means these guys need to be monitored frequently. And luckily, blood draws are cheap. So when he functionalized it, he developed an assay to predict how the drug he was on is working. And this guy was on a drug called bicalutamide, which is an earlier generation anti-androgen. And his test showed that the tumor was turning this drug from an antagonist to a potent agonist. In other words, the tumor was using it as food or fuel, okay? And that's shown here where the drug is is activating the AR pathway. And so this guy was switched on to enzalutamide, a newer generation, more potent drug, and that's what I'm showing you here. Now we see that these two, which were 50-50, dropped to 15 and 5%, and these other mutations emerge, all of which are now converting enzalutamide from an antagonist to a potent agonist, right? So again, this is amazing that the tumors can respond this quickly. And you see that these two here, the original ones, they're still being antagonized. So this is the story of this is really a great thing about the prostate center. We're able to move quickly. So this is a guy, right after we figured all this stuff out, a patient came in. He'd been, this is his PSA, very high, and he was put on bicalutamide and other drugs. His PSA dropped, showing a response, but then started to rise again. When he was, and so at this point, we sequenced his circulating tumor DNA, and again, we found that the bicalutamide, which is also called Casodex, was acting as a potent fuel for the tumor, all right? And he had compound mutations. Another mutation was causing him to convert dihydrotestosterone from its normal agonist activity to a superagonist, right? Now that's shown here. This is response to the receptor to the mutation versus normal. So this guy was in big trouble. He had mets all over his body. He was going to die. And he was switched to the abiraterone, which shuts down production of dihydrotestosterone, and enzalutamide, which antagonized successfully the receptor. And his mets cleared up, and he was well for several months. But eventually he came back and he died. So that's sort of the mutational Darwinian selection aspect of this. But there's something else called lineage plasticity, right? And that means the stem cell for prostate cancer gives rise to luminal and basal cells, okay? And the luminal cells line the lumen of the prostate. And then there's these rare neuroendocrine cells, right? But the thing is that stem cell can give rise to any of those, and does give rise to all of them, right? And so in about 20 to 30% of cases where guys get these potent treatments, they just turn into neuronal cells. And that's terrible because it's lethal quickly. And now, many years ago, I don't know, about 8 years ago, my good friend YZ came running into my office and said one of the patient tumors he had put into a mouse, castrated, which is like treating with these potent drugs, that it had turned into a neuroendocrine. This is the only model in the world. So, we did all the work. We said, yeah, this is really neuroendocrine using immunohistochemistry. And because it's in a mouse, not a human, we can do time course studies on this and see what's the mechanism, how does this happen? And so I won't take you through all these slides in detail, except to say when we did this, we could see a clear transition from one state to the other state. So we did what I like to do, and we sequenced, just assuming this was going to be a Darwinian selection for a subclone, and this would be a great paper, but except that's not what we found. We found that whether we looked at copy number between the patient, the primary xenograft, or the recurrent neuroendocrine cancer, no difference, a copy number. When we looked at fusion genes, no difference. We ruled out contamination because the reviewers made us do that. So this said, well, this isn't clonal selection. This is something different. So what it's called is neuroendocrine transdifferentiation, when it goes from the adenocarcinoma state to a neuroendocrine state. And again, because this is a time course study, we could look at gene expression across this area, and we could identify genes of interest, right? So the androgen receptor drops off as it should because we're castrating and it's turning into a neuroendocrine cancer. Neuroendocrine cancers don't need the AR receptor, so it's gone, right? This other interesting gene, REST, disappeared. What's going on here? And then we had this gene here we call PEG10, which went from 0 to screaming, and another gene that went from 0 to very hot in the neuroendocrine state. And so this led to a lot of research, basic fundamental research, and that I'll summarize quickly here. So what is REST? REST is a neuronal master regulator. And it normally sits on its cognate sites in the genome and makes sure that neuronal genes are not expressed in non-neuronal tissues. And that's regulated by the androgen receptor. Remember, I told you that the stem cells can give rise to luminal and basal cells, which are epithelial cells, or neuronal cells. So AR shuts it off, okay? And when we take out the androgen receptor with these potent drugs, that de-represses a ubiquitin ligase that ubiquinates REST, which then gets degraded in the proteasome. Bang, on go your neuronal genes. So that was interesting. And then, because I dabble in computer science through people smarter than me, not myself, we developed a program or a tool for looking at alternative splicing. And it turned out that other gene I told you about that goes from 0 to high is SRM4, and that's a splicing factor that functionally reprograms a transcriptome, including REST, okay? And again, I won't go into all the details here, but this is my postdoc Nilgan Donmez and Shushen Dong, another PI at the Prostate Center. And what we found, and this has also just been published in European Urology, is that when this gets turned on, it drives neuroendocrine transdifferentiation and is especially potent in the presence of a RB1 and or TP53 loss of function, mutation or deletion. And so this is what's going on, right? So REST normally binds at its cognate sites, turns off all these genes, right? But then this splicing factor comes in and causes it to be spliced to a different function. So REST goes from this state to this state, which is called REST4, which can no longer bind and turn off all these neuronal genes. Okay, so REST, protein stability is regulated by the androgen receptor and isoform switching by SRM4. So this tells us this is a very important gene. There's two big mechanisms keeping a check on it. And so the other gene we found is PEG10, and this was characterized by Shisuke Akamatsu primarily, and my postdoc Alex Wyatt, who's now an assistant professor. Again, you know, we identified it sitting here, PEG10, and this gene was found as being the most highly upregulated gene immediately post-castration. So you kill the androgen receptor, right? This thing comes up screaming. And this shows how that expression increased post-castration, and then again, a terminal differentiation as it moves into this neuroendocrine state. And of course, we validated all this in patient samples and in the xenografts. And so this just shows that the PEG10, yeah, this really gets turned on in neuroendocrine prostate cancer, and that we can see it occurring where you have a adenocarcinoma turning into neuroendocrine. We can see that it goes from a lower state to a much higher state. This was done with our collaborators at Weill Cornell Medical School, Misha Beltran. So this is true. It really happens in patients. And you know, so even a xenograph, because probably some of you are asking, are these xenografts any good? So that we found this in the xenograph, but it turns out that the patient, over 7 years, had exactly the same thing happen, right? When this guy converted from adenocarcinoma to neuroendocrine and then died as a result of it. And this just shows that. So what we think, because the mouse did it in like, I don't know, let's say a year, 6 months, the patient took 7 years. So it might be, this and other evidence suggests that these patient-derived xenografts, if done correctly, act as accelerators. So you can actually predict what's going to happen to the patient way ahead of time. But we have to prove that. So PEG10 is a very interesting gene. First, it's a retrotransposon. It invaded the human line about 120 million years ago. And what we know is that in adenocarcinoma, where you have high AR, right, PEG10 is turned off in all its nefarious isoforms, right? And P53 and RB are typically normal in early stage prostate cancer. But in neuroendocrine, where AR is shut off, PEG10 and its various isoforms get switched on, and that leads to invasion and proliferation, especially in the absence of RB and P53. And another group has published that this same gene also suppresses apoptosis. So a single gene is doing all this, and it's interesting, this is a placental gene. So we also suspect it's suppressing the immune system. So this is a wiring diagram of the state change. I won't kill you with the details, except to say this is a non-mutational mechanism of tumor progression. Okay. So sometimes precision oncology works well, and this is, again, working on these patient-derived xenografts. And this is a friend of ours, actually, who presented with serous ovarian adenocarcinoma. And her husband said, you know, save her life. And we said, we won't be able to, but we'll try. And this just shows that we put her tumor in the mice, and we sequenced her genome. The sequencing predicted for Herceptin and Avastin, and the tumors grew well in the mice until we gave them drug. So she went on combination therapy, and you know, it hasn't changed my life, but it changed her life, because I presented this 5 years ago, same story, and she's 72, running a mile or two every day or swimming, and her markers are stable. But, you know, this is where we're at so far with our maintenance of her, right? Starting in 2011, and or earlier, I guess. And this is a lot of work and a lot of expenses. This is less than half of Becky's team. This is just some of my lab. This does not include the PDX team, which is larger, the surgeons or oncologists in Canada, Israel, and the US, because her husband, James, refuses to give up. So this is not sustainable for healthcare system infrastructures or budgets. That's just not going to happen, right, the way it's happening. So these are my concluding questions and thoughts. So precision oncology is not, in fact, delivering on the hype. In part, it's because there are too few drugs and therapeutic modalities. In larger part, it's because biological complexity cannot at present be predicted from tumor omics alone. I just showed you that the surrounding tissue is somehow facilitating metastasis, and it goes way beyond that. Combinatorial therapies of 3 to 5 drugs are likely needed, but this may introduce unwieldy toxicities. And then should the drugs be sequenced, meaning given sequentially? And if so, in what order? Or should they be delivered in small cocktails? How can this be addressed in clinical trials where enrollees are already limited and costs high? Maybe PDX models and organoids can help. I think that in the future, precision oncology will lead to greatly improved disease management and increased quality of life rather than cures. But in the meantime, it should not be oversold. We're not doing anybody a favor by overselling this. However, to be optimistic, most of us do have cases that give us reason for optimism. So with that, I will stop, thank all my many collaborators, and take questions.